everybody to the first SynBioBeta Deep Dive. We are live here at Amyris in sunny California, Emeryville, just outside of San Francisco. Today we have a very special topic for you where we're going to get deep down and inside the Labcyte Echo. Labcyte is one of the sponsors of SynBioBeta, and you've probably seen Mark Fisher-Corkery, who spoke on stage last year, and Rich Ellison, who's the founder of Labcyte. This new format is one where we can roll up our sleeves and look at some of the details of how this technology platform is going to work. So I'm joined today by Chance Elliott, who's the Director of Automation here at Amyris, and we're going to be learning a bit more about Amyris and what they do and what Chance does and how they use the Echo. And I'm also joined by Jefferson, who's an application scientist at Labcyte, and Labcyte's down in San Jose, about an hour south of here. So we're going to have some Q&A via the webinar, so if you have any questions throughout, then you can just type it into the box. And we have Kevin Costa, who's our live studio audience today, who's going to be taking your questions. We also have a free ticket to SynBioBeta for the best question of the whole webinar, so feel free to send your question and have a chance to win. Labcyte's going to be at SynBioBeta, Amyris is always at SynBioBeta, so I think it's going to be a great event. So Chance, the Director of Automation, has 20 years of laboratory automation experience across a variety of biotechnology instrumentation firms, including Amgen, Cytokinetics, and Agilent. And he spent the last 7 years at Amyris applying industrial engineering techniques to synthetic biology to accelerate the commercialization of renewable chemicals produced from sustainable sources, which is what Amyris is famous for. And he has a BS in chemistry from the University of Colorado. And Jefferson focuses on synthetic biology and next-generation sequencing applications of the Labcyte Echo. Jefferson is a veteran both of Amyris and of Intrexon, and his work on yeast central metabolism appears in Nature. And he has a degree in microbiology from UC Berkeley, which is now growing. So Jefferson, a lot of the SynBioBeta network uses the Labcyte Echo. In your own words, what does this machine do?
So the Labcyte Echo is a liquid handling platform that doesn't use tips. It uses acoustic energy to transfer your liquids from plate to plate. And we find that it's being very useful in scaling down and miniaturizing the assays that are being run, as well as increasing the speed at which people are able to crank through these samples.
Excellent. So I've seen this in action, and one of the beautiful things is the way that it can shoot droplets of liquid from a glass plate into wells on the plate. You've got a new product that just was launched earlier this year. What is that new product?
That new product is the Echo 655T and the Axis 2.0 automation system that goes with it. And it was just launched. We've added a couple new features to the instrument, including the Z-axis motor for the transducer, as well as the ability to use Brooks tubes to, you can basically hit pick the samples that you want from your storage system instead of needing to pull out a whole plate and across that. You can now hit pick exactly what you need and then put it on the Echo.
Excellent. Great. So to begin, Jefferson, you have a few slides that you wanted to talk us through. The first is the main liquid handler.
Yeah. So just to give a quick overview for if there's anybody who's less familiar with the instrument, we produce 3 different models right now, as well as the new 655T that just came out. The Echo 525 is the most popular in synthetic biology. It transfers 25 nanoliters at a time, with a flow rate of about 5 to 6 microliters per second. It's good for transferring master mixes and reagents, as well as precious samples such as DNA. And our other instrument is the 555, and that transfers 2.5 nanoliters. So much smaller droplet size, and it's better for a much smaller type of reactions. And so to give a quick overview on how the technology works, basically we have a transducer that uses a coupling fluid, and We ping basically each well and we listen for that echo. That helps us determine the height of the fluid and how much fluid is in there. We also look for surface meniscus perturbations, and that helps us determine the viscosity. That helps the instrument determine the viscosity of the liquid in there. And so we're able to basically then shoot it with low energy that doesn't really disturb anything in your liquid, and send it to an inverted plate on top. And that's how we transfer liquids.
So I didn't realize, I thought that the beauty of the echo was just that you can use sound waves to actually shoot the liquid into surfaces, but it's actually that you can use a sound wave to analyze all the properties of the droplet as well.
Yeah, the instrument does that before it shoots any liquid. So it's critical to understand what the solution is and how much energy is needed to displace it.
Okay. Excellent.
So quickly, the dynamic fluid analysis is one of our biggest technologies that we've implemented into the instrument. And this, as I mentioned before, we look for surface perturbations to determine viscosity. So the user can have any sort of fluids in there. DNA, anything aqueous, DMSO, glycerol-based solutions, cell lysates, and the instrument will automatically determine the viscosity of that and also calculate the amount of energy needed to eject that droplet. And so using this technology, we can really just remove serial dilutions from workflows, and now we do what we call direct dilution, where we can transfer as little as 25 nanoliters at a time, and that allows us to basically not propagate serial dilution errors, and we can build beautiful standard curves.
Excellent.
And so just wanted to show a couple of pieces. We love supporting our customers and our applications, what people are doing out there, and so a lot of these are available on the Labcyte website. Here we have an application note detailing 5-piece Gibson assemblies at 500 nanoliter scale. And just as an example, we were able to plate, so we can shoot E. coli from a source plate into an inverted Omni tray. So instead of doing your traditional 10 centimeter Petri dish plating, we can now compress that into one Omni tray. Here we have the famous and original Amyris paper, utilizing the Labcyte Echo to do miniaturized, 100-fold miniaturized Nextera sequencing. We've also put out a Nextera XT application note for people interested in using that application. We're supporting lots of new applications such as RNA-seq, as well as microbiome work, 16S rRNA, as well as whole genome sequencing. And lots more coming down the pipeline. So.
Excellent, perfect. In general, what do you think are the kind of customers that stand to benefit most from using the Echo?
Basically, if there's high throughput needs to crank the design, build, learn, test cycle, the Labcyte Echo can really accelerate that by saving a ton of money on the design or the build side of it, as well as speeding up that iteration cycle.
And can you give an example of the cost savings that you can achieve through miniaturization?
Sure. In the Amyris paper, they basically quote that an Nextera reaction, when you do it at scale, it's $72. Being that we're able to miniaturize that 100-fold, we can get it down to 72 cents a reaction.
Excellent. Okay, so we're now going to move over to Chance, and you're going to talk about two topics today. Is that right?
Well, I'm going to give you, first of all, an overview of our Strand construction pipeline, and then talk to you about how we've miniaturized that process and sort of the made us result of that.
Great. Okay, so Chance, over to you.
All right, thanks, John. First of all, just really excited to be part of the first ever SynBioBeta Deep Dive. This is an honor for Amyris. We're very excited to participate. As I mentioned, I want to talk a little bit about the strain construction pipeline at Amyris, and then we're going to go into a little bit of detail around how we've miniaturized many of the steps within that process and sort of the gains that we've realized as a result of that miniaturization. I think the real story that I want everyone to take away from this is one of continuous improvement. For us, that seed started with the miniaturization process, and for the audience members, that might not be the same seed, but the message is the same. You should always be asking questions, always be looking for opportunities to improve your processes. So that's kind of the big picture. I wanted to start out just by giving a broad corporate overview of Amyris, who we are, and what we do for those of you who aren't familiar. So Amyris is about 480 employees worldwide, and we have about 200 of those dedicated to specifically to R&D. We have access to over 2 million liters of fermentation capacity, and we're actually one of the few companies that are capable of taking a product from strain design all the way through manufacturing. That gives us a unique value proposition for our customers. And as a result, right now there are over 1,000 brands that are using Amyris ingredients. So our core business strategy relies on engineering microbial factors to convert sugar feedstocks into a wide variety of chemicals. We then go on to produce those ingredients at scale, and then we go further and commercialize those products either through collaboration partners or by formulating and selling our own products. Biossance is probably the most visible of those product lines right now, and that's one of the fastest growing brands in Sephora right now.
And what is Biossance? What are the kinds of products that
So Biossance is our skincare company, and it's actually a fully owned company of Amyris. They're here, so the Biossance team is here working closely in collaboration with the scientists.
and do you use Biossance?
I absolutely use Biossance. My wife uses Biossance.
I use it as well.
You do?
I admit I do use it.
It's a fantastic. I want to get. It's a fantastic.
I feel left out. I need to use Biossance.
Oh, yeah, we have skin. We got skin cream. We got eye cream. We got sunscreen. Now it's the whole deal. And as I mentioned, it's the fastest growing brand in Sephora, which certainly surprised me.
And so the majority of this is through yeast, the production. Sugar in a yeast, yeast through molecule.
Right. So we feed our yeast sugar. The yeast is going to break those chemical bonds and basically rearrange that carbon into molecules of our choice. In the case of Biossance, that main ingredient is going to be squalane. Right. So just to drive this point home, right now we have manufactured 7 products at scale, and of those 7 fermentation products, we've commercialized 15 products that are a direct result of those 7. And that covers a diverse range of product categories from health and nutrition to clean beauty, which is the Biossance brand, to flavors and fragrances. Kind of going back to this, what we were talking about before, we're doing this by introducing a fundamentally different way of manufacturing ingredients to the world. We feed our yeast sugar. They convert it into products that we normally get either from extracts, animals, or petrochemistry. I think Biossance is an interesting example of this because squalane was originally something that you would get from the liver of sharks. So back in the day, they used to go out and hunt a bunch of sharks, remove their livers, and do an extraction process to get squalane. And it was a really popular ingredient for the cosmetics industry. Not surprisingly, that practice became pretty unpopular, and that was phased out. As a result, that ingredient disappeared as the ingredient for the cosmetics industry. By engineering that into yeast, we were actually able to kind of resurrect this product that had disappeared And now, as a result, it's a really popular ingredient. So this approach allows us to create a product that's better. It's a higher purity profile than you would get from other processes. It has a better cost point, and we're using a process that's sustainable and scalable. All right, so we just talked about how we're making ingredients in a fundamentally different way. Now let's talk a little bit about our R&D pipeline. We've really been focused on accelerating product development. So you'll note that our first product took nearly 40 months from initial strain engineering through production. Contrast that with one of our later products where we're coming in just under 7 months from initial strain design through production. So we've lowered that product development cost, we've lowered that time to market, and we've also increased our own internal product pipeline 6-fold while nominally increasing our headcount. This productivity multiplier is a direct result of our development pipeline. So we've integrated automation, data capture, machine learning, and human intelligence into every aspect of our activities here. Okay, so that was the corporate overview. Now let's talk in a little bit more detail about our Strand engineering pipeline. So just a little bit of background here. Design and construction of DNA traditionally was one of the most labor-intensive aspects of strain engineering. Typically, this would take up to 50% of a strain engineer's time. Thus, DNA design and assembly is a key bottleneck in the strain construction pipeline. That's going to limit our ability to work on multiple molecules at a given time. So at Amyris, our pipeline increases that throughput by nearly 1,000-fold. So we can work on more products and we can reduce that time to market. So in the following slides, I'm going to give you an overview of the strain construction pipeline. So strain construction can be broken down into 4 distinct sub-pipelines with automated strain design as the input. First, we have the parts creation pipeline, and that's where we're going to generate small pieces of DNA. Next, we have the assembly creation pipeline, and that's where we're going to stitch the smaller pieces of DNA into larger assemblies. We're going to use next-gen sequencing to QC the result of the assembly creation pipeline. And then we're going to transform those assemblies into host organisms. So strain construction starts with strain design. And for the purpose of this talk, I'm not going to get into any detail about the automated strain design process. But the short story is that once a metabolic pathway is identified from sugar to target molecule, a set of designs is going to be created. Those designs are going to be translated into GSL code. GSL code is basically a DNA programming language that was developed here at Amyris. That GSL code is going to be submitted to Thumper. Thumper is our manufacturing execution system for this pipeline. Thumper in turn is going to generate a bill of materials for all the parts and assemblies. So all the primer orders, all the templates, all the robotic work lists, etc. So starting with parts. Parts are the most atomic element of our pipeline. So we isolate and amplify individual parts via PCR. Now the sources of these parts, part templates, they could be assemblies from previous development cycles. They could be parts from previous development cycles. It could be genomic DNA, or it could be genes that we've ordered from an external vendor. As you can imagine, doing this at any kind of scale requires a huge number of rearray steps where you're taking templates and primers and mixing them together in reaction wells prior to PCR. So moving on to the assembly step. Once parts have been isolated, amplified, and QC'd, they can now be used to create an assembly. Again, we have another huge rearraying effort that's going to be required to take all those individual parts and pool them into a well for the assembly process. We use yeast homologous recombination for the assembly process. You could use Gibson assembly, you could use Golden Gate, you use yeast homologous recombination. So at this point, it would be really, really costly to allow faulty assemblies to be transformed into a host strain and then screened. So to ensure fidelity of our assemblies, we're going to QC them via next-gen sequencing. And I might jump between NGS and next-gen sequencing. NGS is short for next-gen sequencing. Next-gen sequencing requires a pretty large amount of material for the sequencing process. And for that reason, we have to do an amplification step of our assembly. We use something called RCA to do that, rolling circle amplification. And that's really just a convenient way for us to amplify large assemblies. The main benefit to us is that we don't have to do many preps at this step. It's also one of the only robust ways to amplify large assemblies. So for those of you who aren't familiar with next-gen sequencing, the first step is the fragmentation reaction. And that's going to be where our long piece of DNA is going to get chopped up into smaller fragments of DNA, about 300 base pairs in length. Additionally, adapter sequences are going to be added to each end of those small pieces, and that's going to allow us to add barcodes to those pieces in the next step. And I'll talk a little bit about that. So once the adapter sequences have been added, we can now attach the barcodes via PCR. The barcodes are what allow us to take all of these pieces from all of these samples and pull them together and run them in parallel on the sequencer. And it's this multiplexing of samples that is what makes next-gen sequencing such a valuable tool for this industry. It makes it fast enough that we can use it as a QC step, and it also gives us a path towards an affordable way to do QC. So once the barcodes are added, we actually need to QC that library. So we want to make sure that the fragmentation reaction occurred as expected, and we want to make sure that the PCR reaction occurred as expected. So we're going to be looking at distribution of fragment sizes, and we're also going to be characterizing the concentration of the DNA. Finally comes the actual sequencing run. So all of the fragments are going to be pooled, the library is going to be diluted, and then that's going to be loaded onto the MySeq for a sequencing run. Reads are going to be mapped onto a reference sequence, and this is software that we've developed in-house at Amyris. And the result of that is the depth plot that you see in the lower corner of your screen there. And that depth plot is what tells us whether or not an integration or an assembly occurred as expected. This process is entirely automated, so this depth plot is just sort of a visual for the audience, but these calls are automated. All right, once we've QC'd these assemblies, it's now time to do transformations. We use designer nucleases to do our transformations. What that means is that we have a method of doing really precise multiplexed integrations into a host genome. So that means that in a single transformation, we can take lots of pieces of DNA and integrate them in lots of different loci in a reference genome, host genome. Okay, so the next step is going to be the first step in our actual physical transformation. So you're probably sensing a common theme at this point. We have all of these assemblies in different wells in our sources. We need to rearray those assemblies into consolidated wells for the transformation process. Again, this is a huge rearraying effort that's required to get all of those source wells into their respective destination wells for assembly. Once that, we call this the donor DNA consolidation step. once that donor DNA has been consolidated, it's going to be mini prepped. Now we have and purified. Now we have linear DNA, which is ready for transformation. So the actual transformation step occurs when the donor DNA is added to the competent cell mixture. That's going to be plated onto solid media for selection, and then automated colony picking is going to pick that into culture plates, which will then be used for both QC and downstream strain screening. Finally, transformed strains are submitted for screening. We're going to do a CPCR, which is colony PCR, as our QC method at this point. And that's just a way for us to amplify certain sequences. So we're looking for certain sequences associated with our integrations. And the presence of those sequences in our CPCR process tells us whether or not the integration occurred as expected. This is just a way for us to avoid having to do a whole genome sequence to find out whether we got our integrations at this point. QC failed wells are going to be digitally flagged, and those plates are going to pass through our entire screening process. The results of that screening process, the data is going to feed back into the automated design and build cycle the next time around. So we're constantly learning from our successes and from our failures. Okay, so that was the big overview of the pipeline. Now let's talk a little bit about miniaturization.
So just before we go on, so the overview that you gave is showing that you've got a genetic design, you want to get it inside your yeast cell, and then you want to transform that yeast cell, and then you want to screen for it down the road to see the product that it's producing, correct?
Right. So the purpose of the design, and I didn't talk about the design piece of this anyway, just due to time constraints. The purpose of the design is actually just to actually introduce a pathway into your host organism that's going to channel that carbon path from what yeast wants to do with it to the product that you're trying to make. So that what you're doing in the design process is actually building that root in a organism.
And through the assembly methods that you use, there will be many assemblies that are just wrong and correct, just randomly. So you have this method, you're using PCR to validate which ones are correct, and then you're going to take those, only those, and move them on to the next step of the process. Is that right?
Right. So we want to make sure that the assembly occurred properly. We want to make sure the PCR occurred properly. We want to make sure the assembly occurred properly. And finally, we want to make sure that the transformation occurred properly. That doesn't mean that the design necessarily did exactly what we wanted it to do. So that's all a way just to get these lots of designs into the pipeline so that we can test these designs and allow our machine learning algorithms to kind of deconvolute what designs were good, what designs were bad, and make better designs in subsequent iterations.
Right, so on to miniaturization.
Right, so we talked about the construction pipeline. Let's talk about miniaturization. And this is something that's really allowed us to really think differently about our pipeline and think differently about all of our pipelines at Amyris and realize some efficiency gains and cost savings that just wouldn't have been possible in the past. So how does miniaturization impact the strain construction pipeline? So put simply, lower reaction volumes mean reduced enzyme costs. Non-contact liquid handling means lower tip usage, so we have less consumable costs. Additionally, the Echo is much faster than a traditional liquid or air displacement pipette, and that means our processing times are much faster. And lastly, because the Echo is inherently more accurate, and Jefferson touched on this, you've got the closed loop feedback with the Echo platform, so we can actually detect when a liquid transfer failure happens, and we can flag that and we can respond to that in real time. And as a result, we have a higher success rate. So let's talk some actual numbers here. This is the parts generation process, so this is the first step in the pipeline. We've been able to, through use of the Echo, we've been able to reduce primer costs by 50%. We've been able to reduce enzyme costs by 63%, and we've been able to reduce robot time by 32%. So that's a total savings for a part generation cycle of 47%, and we're just talking about reagents and consumables here. We're not talking about labor. So not only have we reduced the costs, but we've simultaneously increased the quality and increased the capacity. So that's kind of the holy grail. So what you're seeing here is a little bit, it's a little bit busy, but this is a control chart from our part generation pipeline. And basically what I want you to pay attention to are the two solid lines at the top. You see a blue one and an orange one, and then I've got a box around the implementation of the echo-based workflow. So on the x-axis you have cycles, so that's basically time. And on the y-axis you have both PCR success rate and number of reaction levels. And what this is showing is that the blue lines are initial attempts at a PCR run. The orange line is the result of the cumulative result of the initial attempt and the retry attempt. So every time we try to make a part, we're going to try it one time. If it fails, we're going to try to make it another time. So everybody gets a retry. So you'll see at the beginning, first of all, this process from a control standpoint is, what we would define as out of control up to when we start to implement the echo-based workflow. From that point on, you can see that the difference between the residues and the initial attempts really starts to tighten up. So we're getting better at making those assemblies, making those parts on the first attempt. In addition, we're trending upwards. That gap is narrowing, and where we are today, we're nearly 100% success rate on our PCR process. So additionally, assembly generation. So we talked about parts, let's talk about assembly generation. One of the key bottlenecks in the assembly generations process is the part mixing step. This is another big rearray process. This used to be a multiple robot, multiple day rearray using a traditional air displacement liquid handler type of setup. We've reduced that by 80%. This is something that we can now do on one machine in a matter of hours. This is the next generation sequencing step. This is Jefferson talked about the paper that we produced. That original Nextera protocol came in at $82 per reaction. So for a pipeline like ours, where we're literally making thousands of constructs on a weekly basis, $82 a reaction is not a realistic way to do QC. But next-gen sequencing is really the only way you have of ensuring the fidelity of your constructs. Through a grant from DARPA, we were able to miniaturize this reaction. And at the time of the publication of the paper, we had reduced this to a half microliter reaction. We're at a little over $2.50 per reaction. As Jefferson mentioned in his talk, this has, we've continually driven this cost down by further miniaturization. Moving on to transformation. Again, the donor DNA consolidation step was a massive rearray process. And similar to the assembly part consolidation step, we took a process that was running on multiple robots over multiple days and reduced it to a single robot, hour-and-a-half process. And that's a really big deal. If you think about a pipeline effort where to do this for multiple steps, you got multiple robots that you need up for multiple days. So you lose a robot, not only have you broken this part of your pipeline, but you've broken upstream and downstream parts of your pipeline. And now your schedule starts to slip and really everything falls apart. So you need to find some way to create space to allow for failure events to happen. You can't do that when you're leveraging all of your robots to do all these processes. So this was really a sea change for us to be able to miniaturize these reactions. And so next steps. So we really feel like we're just kind of scratching the surface as to where we can apply this technology. From solid media plating, which Jefferson alluded to dot blot assays, antimicrobial testing, mass spec direct injection. Really, the sky's the limit in terms of sorry, 2 different slides.
Sorry about that.
Really, the sky's the limit in terms of where we can apply this technology. Okay, so just to summarize, I've shown you all the steps in the strain engineering process. I've talked about the impact of miniaturization at each step. So let's talk about the bottom line. So the bottom line, part generation efficiency, we're seeing a 20% increase in efficiency there. Assembly generation efficiency, we're seeing a 20% increase there. Capacity, we're seeing a 50% increase there. And the chart you see up in the corner actually shows functionally we utilize a 50% increase in capacity. That chart in the corner there is a CPCR, which is sort of a rough surrogate for our total pipeline capacity. And that's an area where we're seeing 104% increases year over year. So that 50% increase, we're not even scratching our capacity at that point. In addition, 30% reduction in cost just off the top. And again, we're not talking about labor. So just let that sink in for a minute. We've increased capacity, we've increased efficiency, and we've decreased the cost. So the cost of a cycle that was less efficient, running at less capacity, was more expensive than the cycles that we run today. Okay, so I just want to acknowledge that this was originally built as a live demo, and I don't want to leave the audience wanting. So what you're seeing here is a time lapse of our dual echo platform. We affectionately refer to this as the echo chamber. This is doing a PCR setup process, and you can see with all of the swapping of the plates, just how intensive these rearray efforts can be, and how much speed you can realize when you miniaturize these processes. So going back to my original statement, even if miniaturization isn't the answer for you, I hope what I've shown will inspire you to kind of rethink some of your own processes, ask critical questions, constantly be looking for improvement opportunities. So with that, I'd like to thank the audience for their time. Thank you for your time, and acknowledge all the hard work from the people who really made this possible. So our fantastic automated strain engineering team, who are constantly looking for new ways to improve the processes, our bioinformatics and software engineering group, who allow us to drive all of these processes, allow us to capture all the data and learn from all the results of that data. And then, of course, my team in automation engineering, the talented people who do the hard work that make my job look so easy. Lastly, we're hiring. So if you care about sustainability, if you care about the way the ingredients you use are produced, and if you're passionate about science and engineering, come join us. We'd love to meet you. Thank you.
Fantastic. Excellent. Thank you, Chance. So I'm very curious as to some of the questions that we have online. I see that we have a few that have come up here. I'm just going to go back to the screen here. Kevin, do we have the first question?
Yeah, absolutely.
So hold on one second. We're just going to pass the microphone over to Kevin.
Sure. So Jen Wang asked, how do you analyze the target chemicals in the engineered yeast strains in a high throughput manner?
Great question.
That's a great question, and I wish that I had an easy answer for you. I don't think that There isn't a great generic way to do this sort of analysis. I would say that probably the most generic tool that we use is mass spec, but as a high throughput process, there are some technologies which we have in-house that allow us to do that in a high throughput setting. But I think that's one of the areas where I would love to see more innovation is some more generic ways to measure these products.
Jefferson, maybe you can tell us a little bit about some of the future products that are going to be coming down the line from Labcyte. In particular, you've got a mass spec tool.
Sure. We've started to release some news and some alpha prototypes of an echo transducer coupled to a mass spec, and by firing these femtomole or femtoliter droplets, we're able to analyze 3 samples per second on acute health. That's about probably 2 years out from being able to be commercial scale.
That's huge.
Yeah, that brings up an interesting point about integration. I presume that there's a range of implementation partners that you work with. Can you explain a bit about that? If somebody wants to try out the Echo, what do they do? And then if they want to produce an echo chamber or something equally as cool for their lab, what's the process there?
So we have a pretty global and strong applications team, and if somebody's interested in trying the Echo out, we can do typically a 1-week demonstration where we fly on an instrument and an application scientist. We work on the workflow with the customer or potential customer, and produce some data and see how the Echo can increase the quality of data as well as decrease the cost and the time required to run these workflows.
Excellent, great. Kevin, do we have another question?
Yeah. So Tong Zi says that the use of Echo for single colony plating on OmniTray is awesome. And he asks, is there an application note or a research paper published about that process?
We haven't published anything on that process.
We should have an application note on the website, labcyte.com. If you look at application notes, G125 is the number of that application note, but it is detailed there.
Great. Jefferson, I was reading on the Labcyte blog recently about the way the Echo is being used to improve animal research for bioproducts. I wonder if you could give us a bit more detail on that.
Sure. So a little bit of background on the Echo. Labcyte's been around since 2004, and that's where we began. Was a lot of DMSO-based solutions, and a lot of drug and pharma testing, screening for drug analogs. Because of the small droplet size, we were able to really save these precious materials and test a ton of different analogs. So in traditional models, there's a lot of animal models that are required to pass these drugs forward. By using the Echo, these pharma companies are able to really drastically reduce the amount of animal models that are needed just by size reduction and speed reduction.
Excellent, thank you very much. So the Q&A is still open, so if you have extra Q&A that you want to ask, then please just type it in the box on Zoom. I want to know, Chance, how what the lab scientists are doing with their extra time now that the Echo is speeding up what they do.
So, right,
Where have you laid them off?
we definitely have. We're growing. We're not laying anybody off. So, I mean, I showed in some of those original slides how we've managed to decrease time to market and increase our product portfolio without adding a bunch of headcount. So that's how these efficiencies, that's how we're utilizing that. So we are growing slightly, but we're not growing at the same pace that we would need to traditionally support these new efforts. So we're utilizing that time by basically adding a lot more products to the portfolio, doing a lot more development in parallel.
And do the reagent manufacturers hate you because you're buying now 70% less reagents, fewer reagents than you were buying before?
That's a great question, and I certainly can't comment on the disposition of some of our suppliers. Curiously, there was a release that just went out from Illumina that they are phasing out the Nextera XT kit, and that is the one that allows us to do this miniaturization. So that's something that Labcyte and Amyris needs to have a discussion about.
I've heard that announcement as well, and Illumina has a new bead-based kit, and so I'm sure we'll be able to continue to innovate on that front with their new products and continue to keep this pipeline as cost-effective and efficient as possible.
We'll figure out a way to make it cheap.
Can you go back to the when you first started using the Echo, and how has it, now that you've got it, when we come up to do a new automation protocol, how has it changed your thinking in terms of how you might design an experiment or how you might gather the data? It's a fundamentally different tool, and it allows you to do things at a different scale than you've ever been able to do it before. So how does that change either your thinking or the lab scientists?
Well, in terms of, I think it is the tool that has allowed us to scale the pipeline. That's kind of where we were maybe 2 years ago, of just really sitting there thinking, how are we going to double, triple the pipeline capacity, and really needing to search for a technology that would support that kind of growth. So probably not so much around the design of experiment side of things, but just looking at how can we miniaturize all these processes? How can we take everything that we're doing right now on what we would call an advanced liquid handler, a traditional span 812 style air displacement pipetter? How can we take all those processes and miniaturize them and leverage the Echo for all these processes?
I see a lot of opportunity in enabling certain workflows that were too costly in the past. For example, for immunogenesis pipeline, you can now routinely whole genome sequence the strain to find out what mutations occurred after you transformed the strain, if anything weird happened across the genome. And people are looking into CRISPR interference, CRISPR-I, CRISPR-A, to do genome-wide scale knockdowns or activations of certain pathways. So by miniaturizing the reaction, you can really test the entire genome space at a cost-effective, time-effective scale.
Excellent. We have another question online from Tom Si. And if you're asking a question online, maybe just let us know which company you're at or which institution you're at or where in the world you're at. It'd be interesting to know. And Tom asks, for amplification of yeast plasmids using RCA, is boiling or Zymolase used to break up the cells?
Yes, so I didn't talk in detail about the kind of molecular biology steps behind it, but yes, absolutely. You have to boil the cells prior to proceeding.
Great, excellent. So I want to talk a little bit about Synthetic Biology Week, which is happening at the end of September, the first week of October. It's the week of SynBioBeta, and there are a couple of workshops happening down at Labcyte in San Jose. So if people go to the SynBioBeta website or syntheticbiologyweek.com, you can see there's over 25 events that are on the website. We're going to be launching those next week. So if you want to sign up, they're all free. And you can actually go down to the Labcyte lab. Actually, we might be doing it at UCSF now. And you can and you'll be able to get a hands-on demo and take a workshop in using the Echo yourself. Great. So if we have more questions online, then please send them in. We have some examples, some free tickets to SynBioBeta to give out for the best question. I got another question that we thought of before, which was, can you describe direct dilution and why that's better than serial dilution? What's it used for? And who might benefit from it?
Sure. Direct dilution is what we call an echo process, where traditionally, if you were to build a standard curve, you would do serial dilutions. You'd have your high concentration standard and dilute it down tenfold, hundredfold, whatever you need to build out your standard curve, and transfer equal parts volume of each of those dilutions. And it's well known that there's a lot of serial propagation errors that could occur. Even within the tips, you can have your standards stick to the tips, mixing issues. It's a very fragile process. And so by using direct dilution, we basically are able to shoot at 25 nanoliter or 2.5 nanoliter increments the amount of standard that you need. So without using tips, so we're able to basically transfer from one source and create your entire standard curve in 25 nanoliter increments to hit the targets that you need. And we generate very tight standard curves using that process. In pharma, it's a big deal because people are able to just dose in the drug analogs and build up from that without worry of potential other variation effects of like pipetting or using material elsewhere.
Excellent. Thinking about how we talk a lot about DNA and droplets of liquid with DNA for assembly. Can cells safely be transported by acoustic energy, and what's the effect on normal cell function if you try to put cells through the echo? Do you do any of that?
That would be analogous to the plating function. So we're plating live culture onto solid media.
Okay, apologies.
Yeah, as far as we've seen, we haven't had any effects on cells. It's very low energy. We've also been asked whether transferring the droplets may shear the DNA, if there's any sort of force there, and we haven't seen that. And we've tested that. It's less energy than the acoustic force used in the Cumbers. It's much less. And so we're able to plate cells and recover them and pull the DNA, the plasmids back out without issue.
Excellent. Great. Well, if no more questions online, then I think we'll wrap up. Any final comments, Chance?
No, just thank you for your time. Thanks for the audience participation, and my message is just continue to look for opportunities to innovate and never let your processes stagnate.
Jefferson, any final thoughts?
Get in touch with us if you guys are curious, and we'd be happy to talk to you and potentially set up a demo and whatnot. And yeah, thank you guys. Thank you SynBioBeta. Thank you Amyris for having me here and giving us the opportunity to interact and talk to you. It's been great.
Great. And if people do want to schedule a demo, what's the best way they can get in touch with you?
We have local account managers. You should be able to just email any of the Labcyte emails. It will be directed to your local account manager, and then that person will take over and carry the process forward from there.
Great, and let them know that you heard about it first here. SynBioBeta Deep Dive. And we just have a note from Tom. He says he's at the University of Illinois, and he has one last question on the LIMS system that Amyris used to interface Echo and traditional liquid handlers. What was the LIMS system that you used?
You're probably looking for a commercially available system, and I would love to offer that to you. Everything, all the LIMS that we have in-house at Amyris is home built. So these are systems that we built ourselves to try to build systems.
Great. Excellent. So next week we are going to be live from Paris. I'm doing a webinar with City in London And that's going to be all about the global developments and business opportunities around synthetic biology. So that's going to be a little early for us folks in California next week, 6 a.m. Pacific. But if you want to join us, it'll be a fun time and an interactive session where we'll be going over some of the trends that I've been seeing in the industry and some of the business development opportunities that are out there. We have a number of SynBioBeta Lives coming up throughout the year. We have Opentrons. We have one with Ginkgo Bioworks. We have one with Kevin. Who else do we have coming up?
Oh gosh, put me on the spot here.
Anyway, sign up for our newsletter and you'll receive the dates for when those are coming out. So thanks everybody for joining. Thanks again to Jefferson Live from Labcyte and for Chance Elliott from Amyris. Thank you all. We'll see you soon.